Hello future MDs! In this video, we're going to answer the biology part of NMAT Practice Set 2014. This is part 1 of our bio review, where we will answer numbers 1 to 25. But first, let me introduce to you our lecturer. Her name is Angela A. Arma. She graduated cum laude under Bachelor of Secondary Education, major in Biology, minor in Chemistry at the University of the Philippines, Diliman. She took the NMAT last October 2019 and got a percentile rank of 98. And right now, she's studying medicine at the University of Northern Philippines. Nag-self-study lang din daw siya, guys. So I hope you get inspired na mag-aral at maging confident kahit hindi kayo nakapag-review center. If you know someone who needs NMAT reviewers, feel free to share this channel. Also, if you find value to the contents here, please click the subscribe button below. Thank you so much, and let's start.
Question number 1. Chromosomes become most distinct during cell blank. So to answer this question, let us first define what is a chromosome. A chromosome is a long, thread-like structure composed of DNA and proteins that carries the genetic information of an organism. So based on this definition, what cellular process do you think chromosome is the most important? Since it carries the genetic information of an organism, this of course is essential in cell reproduction or division. So the answer here is option A. As what you can see here in the picture, during interphase, the chromatin within their chromosomes is less tightly packed. So this looser configuration is important because it permits the transcription to take place. Then during mitosis or cell division, they become more tightly packed and their condensed form can be visualized with a light microscope. So here is an example image. Question number 2. In which pair of organelles are light and chemical energies transformed? The only organisms that can transform light energy are plants, algae, and some bacteria. That is because they contain organelles called the chloroplasts that carry out the process photosynthesis. So as a review, photosynthesis is the process by which plants, algae, and some bacteria use the energy of sunlight to drive the synthesis of organic molecules such as glucose from carbon dioxide and water. So the byproduct here is oxygen. In the options, only option B and C has chloroplast in the organelle pair, so we can now rule out A and D. Now, which organelle among lysosome and mitochondrion can carry out chemical energy transformation called the cellular respiration? The answer is the mitochondrion. That is why it is called the powerhouse of the cell or the energy factory of the cell, because it is responsible for making ATPs. So the answer here is chloroplast and mitochondrion, which is option C. It is also nice to know where the process of cellular respiration takes place in the mitochondrion. So for glycolysis, it occurs in the cytoplasm. The Krebs cycle or the citric acid cycle occurs in the matrix, while the oxidative phosphorylation occurs in the inner mitochondrial membrane. Question number 3. Which of the following is the function of the nuclear membrane? So here is an image of a nucleus with its nuclear membrane. In this question, your first clue is the word membrane, which means that it envelopes or surrounds something. The other clue is the word nuclear, which means that it's associated with the nucleus. Combining this, we can therefore say that the function of the nuclear membrane is to envelope or surround the nucleus. So that is option C. Remember that nuclear membrane is a double membrane surrounding the nucleus, and it also contains pores called nuclear pore, which regulate the entry and the exit of materials between the nucleus and the cytoplasm. So let's discuss now the other options. Option A, it controls the activities of the cell, is an obvious function of the nucleus. Option B, it encloses the cytoplasm, is a function of another membrane, the plasma membrane or the cell membrane. Option D, it supplies energy for the cell, is a function of the mitochondrion or the powerhouse of the cell. So there are also things I would like you to take note or to remember. The organelles that are bound by double membranes are the nucleus, the mitochondrion, and the chloroplast. And the other organelles with their own genomes are the mitochondrion and the chloroplast. Because of this information, the endosymbiotic theory posits that the mitochondrion and the chloroplast are both organelles that were once free living cells. So it is nice to know this because they sometimes appear on the exam. Number 4. The chemical responsible for most synaptic transmission in neurons is the blank. To answer this question, let us take a look at the mechanism of synaptic transmission. As what you can see here, most neurons communicate via neurotransmitters at the chemical synapses. Acetylcholine is the most abundant neurotransmitter in the peripheral nervous system. So the answer to our question here is option D. Acetylcholine is synthesized in the cytoplasm of the actin terminal from two substrates, the acetyl-CoA and the choline. Acetylcholine is then stored in synaptic vesicle until an action potential causes its release via exocytosis. Acetylcholine then binds to receptors called the cholinergic receptor, and it is degraded by acetylcholinesterase. So let's now discuss the other options. Adrenaline and noradrenaline are similar chemicals that act as both neurotransmitters and hormones. They do not account to most synaptic transmission, unlike the acetylcholine. Choline, as what we have discussed earlier, is just a precursor of the neurotransmitter acetylcholine. For question number 5, which stages of cellular division have exactly opposite characteristics? To answer this, let us review the stages of mitosis or cellular division. So for the first event, which is called the prophase, the nuclear membrane breaks down and the DNA condenses to form chromosomes. In the second event, the newly formed scaffold of spindle tubules attach to chromosomes at the centromere, and the chromosomes are maneuvered into the equator, or what we call the metaphase plate. In the anaphase, the spindle tubules contract and pull the chromatids apart towards the opposite poles of the cell. Lastly, in telophase, the nuclear membrane reforms, the spindle tubules break down, the chromosomes decondense and disappear, and also the cell undergoes cytokinesis. As what you can notice, there are events in the prophase that are exactly opposite of those that are in telophase. For example, the nuclear membrane breaks down in prophase, while in telophase, nuclear membrane reforms or reassemble. In prophase, the chromatins condenses to form chromosomes, while in telophase, the chromosomes decondense and they disappear. So the answer here will be option A, prophase and telophase. And this will be the summary again. Prophase, nuclear membrane breakdown; telophase, nuclear membrane reformation; prophase, chromosome condensation; telophase, chromosome decondensation. Question number 6. The continued synthesis of protein requires the continued synthesis of its corresponding blank. To answer this, let us have a review first on the central dogma of molecular biology. Looking at this diagram, in DNA replication, a double-stranded DNA molecule is copied to produce two identical DNA molecules, so it's DNA to DNA. In transcription, the information is transferred from one strand of the DNA to RNA by the enzyme RNA polymerase. So it's DNA to mRNA or messenger RNA. Lastly, in translation, mRNA is translated into proteins with the help of ribosomes, ntRNA, or transfer RNA. Now, let's go back to the question. So for the continuous protein synthesis, which of the following should be synthesized continuously as well, based on the diagram? Since mRNA codes for the proteins, it is a requirement for protein synthesis in translation process. Question number 7. Which of the following results in the complete oxidation of a substrate to ATP and carbon dioxide and water? So let us first discuss the first option, which is glycolysis. Glycolysis process is shown in this diagram. As what you can see, It came from the word glyco, meaning sugar, and lysis, meaning breakage. So if we will combine this, the simple definition of glycolysis is sugar breakdown or sugar breakage. Glycolysis is a series of reactions that extract energy from glucose by splitting it into two 3-carbon molecules called pyruvates, as what you can see here. Glucose, undergoing the process of glycolysis or splitting down of sugar, will result into two 3-carbon molecules pyruvate. And then in the process, ATP is released. So based on this diagram, as what we can see, it does not satisfy this equation. So we can now rule out glycolysis. Okay, now we can proceed to fermentation. So fermentation is an anaerobic pathway, meaning it does not require oxygen, and it can be an alcohol fermentation, which produces ethanol, or it can be lactic acid fermentation, which produces lactate. Again, just like glycolysis, its product does not equate with this one. Okay, let us now proceed to the third option, which is aerobic respiration. It is called aerobic respiration because it requires oxygen for the reaction to occur. So its main events are glycolysis, Krebs cycle or citric acid cycle, and the electron transport chain or the oxidative phosphorylation phase. So as what you can see here, its products are ATPs, carbon dioxide, and water. So to see it more clearly, here is the chemical equation of the reaction. So as what you can see here, its substrates are glucose and oxygen. And then after undergoing cellular respiration, the products now are carbon dioxide, water, and ATP. So the products here are similar here in our question. So the answer now is aerobic respiration. Question number 8. Enzymes are important to the life of cells because they blank. So let us first define what are enzymes. Enzymes are biological catalysts or biocatalysts that speed up biochemical reactions by lowering the energy of activation. So activation energy is the minimum energy that must be acquired by a molecule to undergo a chemical reaction. So what is the importance of lowering the activation energy? So lowering the activation energy greatly increases the probability that a reaction will occur. So as what you can see here, in a reaction that has no enzyme, the activation energy is very high. While in a catalyzed reaction or in a reaction that utilizes enzyme, the activation energy is significantly lower. So because of this, the probability that a reaction will occur is higher here in a catalyzed reaction. So the answer here is option A. Enzymes speed up chemical reactions in the cells. The succeeding options are not true regarding enzymes. And also, I would like you to take note that enzymes are not consumed during the reaction they catalyze. So, enzymes convert substrate to products while remaining unchanged themselves. And again, it is very important to note that enzymes catalyze reactions by lowering the activation energy. Question number 9. Plant cells will not break when placed in a container with plenty of water due to the presence of blank. So to answer this, let's take a look at a plant cell. Plant cell can be distinguished from an animal cell due to the presence of the following organelles. First, the chloroplast. Second, the large vacuole, which contains water and dissolved substances and functions in maintaining pressure against the inside of the cell wall. And lastly, its cell wall. So, what are cell walls? So, cell wall surrounds the plasma membrane of the plant cells, and it provides tensile strength and protection against mechanical and osmotic stress. It also allows cells to develop turgor pressure. So, a turgor pressure is the pressure of the cell contents against the cell wall. So, for this question, the answer is option B. The cell wall supports the cell membrane when it is turgid. Question number 10. Which of the following is true about catalyzed reactions in cells? Okay, so let us first have a review about enzymes. Remember that enzymes catalyze reactions by lowering the activation energy, and enzymes are not consumed during the reaction they catalyzed. So as what you can see here, the activation energy in an catalyzed reaction is higher, while the activation energy in a catalyzed reaction is lower. So for option A, the catalyst itself becomes involved in the reaction. This one is false because as what we've mentioned earlier, enzymes are not consumed during the reaction. They remain unchanged. And enzymes do not appear in the overall stoichiometry of the reaction it catalyzes. For option D, enzymes cause uniform reaction is also false. For option B, the catalytic efficiency of enzymatic reactions is extremely high. This one is true because as what we've mentioned earlier, lowering the activation energy greatly increases the probability that a reaction will occur. And also, because of its lower activation energy, the reaction rate of the catalyzed reaction is faster than the reaction rate of the uncatalyzed reaction at the same temperature. So that will yield into a higher catalytic efficiency. So the answer here is option B. Question number 11. Which activity does not require cellular energy? So it's asking for an event that does not require ATP. Or, in other words, it's asking for a passive event among these options. So when you see this type of question, the magic word here will be diffusion. If you see diffusion, that would automatically mean that it's a passive event, passive transport, and it does not require energy currency or ATP. This because diffusion, it involves the movement of material along a concentration gradient. So it's from a higher concentration to lower concentration. Example of this is diffusion or simple diffusion and defacilitated diffusion. So some molecules such as carbon dioxide and oxygen, they can diffuse across the plasma membrane directly, but others need help to cross its hydrophobic core. So in facilitated diffusion, molecules diffuse across plasma membrane with the assistance from membrane proteins such as this. For example, here is a channel and here is a carrier protein. So to answer this question, diffusion of sodium chloride through blood plasma is the activity that does not require cellular energy. Question number 12. Which feature is present in eukaryotic cells but not in prokaryotic cells? So here is a table summarizing the differences between prokaryotes and eukaryotes. So in option A, chromosome, actually both prokaryotes and eukaryotes have chromosome. They just differ in their shape or their arrangement. So in prokaryotes, they have single circular chromosome, while eukaryotes have multiple linear chromosomes. And both prokaryotes and eukaryotes have plasma membrane or cytoplasmic membrane. They just differ on the composition. While, as what we can see here, when it comes to their nucleus, eukaryotes have a true nucleus because it consists of nuclear membrane and nucleoli, while prokaryotes do not have true nucleus and they are just called nucleoid because they do not have nuclear membrane. So here is an image of a bacterial cell structure or a prokaryote with a nucleoid DNA. So nucleoid, meaning nucleus-like, is an irregularly shaped region within the cell of a prokaryote that contains all or most of its genetic material. So in contrast to the nucleus of a eukaryotic cell, it is not surrounded by a nuclear membrane. So take note that there are resources that says that nucleolus is also not present in prokaryotic cells unlike eukaryotic cell. So if you will take that in consideration, the two answers here will be nuclear membrane and nucleolus, but we have to choose the best answer, so that would be option C. Question number 13. The growth of lateral buds apparently involves the balance of which three hormones? So to answer this, let us first define what is a lateral bud. So axillary or lateral bud is formed at the junction between a leaf and the stem. So another important to define here is the apical bud, which is, on the other hand, the primary growing point located at the tip. So apical bud is the main area of growth in plants. So for us to understand the growth of the lateral buds, let us first discuss a very important concept, which is the apical dominance. Apical dominance is the ability of the apical bud to suppress the development of the axillary buds. As what you can see here in this plant, which has an active apical bud, the lateral buds are dormant. So most of the growth occurs here in the apical bud. Compared to this other plant, which has no cut apical bud. As what you can see, the removal of the apical bud enables lateral branches to grow. So what's the reason for this? So in plants, the apical bud is a preferred sugar sink and a major site of auxin biosynthesis. So IAA is just auxin. So auxin is a plant hormone that has long been known to inhibit lateral bud growth. Going back as what we've said earlier, in plants with intact apical bud, most of the sugars are concentrated in the apical bud or at the tip. Also, as what you can notice here, auxin moving downward from the apical bud where it is synthesized produces strigolactones that repress the growth of the lateral buds directly. The opposite of this enzyme is the cytokinin, which on the contrary promotes the lateral bud growth. So apical dominance is suppressed under the effect of high cytokinin, and high cytokinin permits the development of the axillary buds. Now, when you cut off the apical bud, just like in this image, you also remove the sugar demand of the apical bud and now rapidly increases the sugar availability to the axillary buds. Removal of the apical bud actually allows the lateral buds to receive more sugars, that's what you can see here, for their growth. Also, it is important to note that the level of auxin, particularly near the cut surface, declines, and this decline allows the topmost axillary buds in particular to grow and now take over as the new apical bud. So I hope that's clear. So going back to the question, which hormones are responsible for the regulation of lateral growth? So we've discussed two so far. They are auxin and the cytokinin. So in our options, only option A and B have auxin and cytokinin in them. Hence, we can now eliminate options B and C. The question now is it gibberellin or ethylene? Okay, so to determine which of these two is the correct answer, let us review first the functions of these hormones. So, gibberellin stimulates the breakdown of stored food to facilitate germination and sprouting of leaves. Ethylene, on the other hand, is responsible in the ripening of leaves and fruits, and it also responsible in the dropping of leaves and fruits. So let us take a look at their role in the apical dominance and the lateral growth. So between these two, if you will look it up on the internet, ethylene has a more established or more defined role in the apical dominance than gibberellin, though there are studies that says that in pea plants, gibberellin actually seems to play an inhibitory role in the lateral bud growth. So we cannot eliminate gibberellin. We will just focus on the best answer, which is ethylene. According to the Encyclopedia of Raw Science in 2003, for auxiliary bud growth regulation, a balance between cytokinins and abscisic acid or inhibitory complex is also possible, as is in interaction with other growth regulators such as ethylene. So what are the specific roles of ethylene in the apical dominance? Ethylene is actually involved in two discrete mechanisms in the control of apical dominance, which of course also controls the growth of the lateral bud. The first one is the release of lateral buds from inhibition and also in their growth and development. So generally, high levels of ethylene in the apical region are actually conducive to lateral bud outgrowth, while high ethylene levels in the region of the lateral buds themselves tend to be inhibitory. So it actually depends. But the best answer here in our options is option D. The hormones responsible for lateral bud growth regulations are auxin, cytokinin, and ethylene. Question number 14. Which of the following are present in both prokaryotic and eukaryotic cells? So let's go back to the table earlier. So for option A, lipid protein cell walls. So cell walls are usually present in prokaryotes. In eukaryotes, they are not present in vertebrates, but they are present in plants and also in fungi. And when they're present, they are chemically simple, unlike those in prokaryotes. So that is not true. For option C, distinct nuclear envelopes. So as what we've mentioned earlier, prokaryotes do not have nuclear membrane, unlike the eukaryotes, which have a true nucleus. Option D, circular chromosomes of DNA. Again, as discussed earlier, the circular chromosomes are only present in prokaryotes because in eukaryotes they are multiple linear chromosomes. So the answer here now is option B, cytoplasmic and nuclear regions. Because what we can see here, cytoplasm is present in both prokaryote and eukaryotes, and they both have nuclear regions. As in prokaryotes, they are called the nucleoid. Question number 15. Which of the following correctly describes the sodium and potassium ions across the cell membrane? So as what we can see here, the sodium ion concentration is lower inside the cell than the outside, and the potassium concentration is greater inside the cell compared to the outside. So this concentration difference for sodium and potassium are due to the action of sodium-potassium pump or sodium-potassium ATPase, which pumps 3 sodium out of the cell and 2 potassium into the cell. As what you can notice, since the movement of ions are against their concentration gradient, that is from low concentration to high concentration. For example here, low concentration of sodium from inside the cell out to a higher concentration, which is here outside, extracellular fluid, and then here potassium, for example, lower concentration here outside to inside of the cell in the cytoplasm, which it has higher concentration. So it is an active transport, and it requires hydrolysis of ATP. So take note that for 2 potassium that will enter the cell, 3 sodium ions exit the cell. So a useful mnemonics for this is PISO, potassium in, sodium out, so that you will not forget this. So just remember PISO, potassium in or 2 potassium in and 3 sodium out. So the answer in our question is the concentration of sodium ions outside the cell is high, and the concentration of potassium ions outside the cell is low. Question number 16. What is the correct sequence of the different stages of blood clotting, or what we also call the process of hemostasis? So hemostasis is the process by which the body seals a ruptured blood vessel to prevent further loss of blood. So the first step in this coagulation cascade is the release of clotting factors from the platelets. So these clotting factors then cause the platelets to become sticky so that they can adhere to the damaged region and form a platelet plug, as what you can see here. Also, clotting factors trigger the conversion of the inactive zymogen prothrombin to the activated enzyme called thrombin. Thrombin in turn catalyzes the conversion of fibrinogen, of the soluble fibrinogen into an insoluble fibrin. And then, the fibrin strands form a mesh of fibers around the platelet plug and traps red blood cells to form a temporary clot. So going back here to our question, what will be the correct sequence now? So the first event will be the rupture of the platelets, so that there will be a release of the clotting factors. And then the next will be the conversion of prothrombin to thrombin, and then followed by the conversion of the soluble fibrinogen to the insoluble fibrin catalyzed by thrombin. And then the last step will be the fibrin plus erythrocytes or RBCs formation of a hardened clot or a temporary clot. So that would be option C. Question number 17. Which of the following is most likely to occur when a cell is placed in a hypertonic solution? So let us first describe what a hypotonic solution is. So a hypotonic solution is a solution with the lower concentration of solutes, and because of that, the water moves in the cell. As what you can see here, the water moves into the cell, and this will cause the animal cell to swell and then eventually burst. So that will cause the lysis of the animal cell. While in plant cell, it will make it turgid, which is actually normal in plant cell. In isotonic solution, where there is an equal concentration of solutes, there will be no net movement of water. So, as what you can see here, the water goes in, the water goes out. So there will be no net movement of water. So the animal cell will retain its normal size, while the plant cell will be flaccid, as what you can see here. Lastly, in a hypertonic solution, where the concentration of the solute is higher compared to the solution of solutes inside the cell, the water tends to move outside. So as what you can see here, the water moves outside from the cell to the solution. Then it will make the animal cell shrivel or plasmolyzed, as in the case of the plant cell. So the answer now to our question is plasmolysis. Question number 18. The function of manufacturing is common in which of the following groups of organelles? So manufacturing meaning their function involves synthesis of a certain product or certain products. So what I did was I listed down here the functions of the organelles included in our options. And let's now do the process of elimination. So for our first group, it includes microtubules, the mitochondria, and Golgi apparatus. Now let's review each of the organelles' function. Microtubules, it's mainly for cytoskeleton and regulation of cell growth and movement. So it's not for manufacturing. Mitochondria for cellular respiration, so you generate energy here, right? In Golgi apparatus, you synthesize many polysaccharides, so it's for manufacturing. Its function is also for manufacturing. However, since in this group, microtubule actually do not function in manufacturing a product, this group therefore is not correct. So we can now eliminate this. For option B, we have chloroplast, lysosomes, and endoplasmic reticula. So let's find out if they have a common function in manufacturing. Chloroplast is for photosynthesis. So in plant cells, chloroplasts manufacture or synthesize sugar or glucose during the process of photosynthesis. So yeah, its function is for manufacturing sugar. Lysosome, on the other hand, is for breaking down ingested substances, and it's mainly for recycling. So this does not function in manufacturing. Endoplasmic reticula, actually, the smooth endoplasmic reticulum functions in the synthesis of lipid. So in this group, the only organelles that function in manufacturing are the chloroplast and the endoplasmic reticulum. So lysosome is for breakdown. This made this group now wrong. So we can eliminate option B also. Option C, Golgi apparatus, cell membrane, and chloroplast. So we already mentioned earlier that Golgi apparatus and chloroplast both function in manufacturing a certain product. In Golgi apparatus, it synthesizes many polysaccharides. In chloroplast, it synthesizes glucose. However, cell membrane, it mainly functions as a protection or a barrier. So it does not manufacture products. So this made this group now wrong. We're left with option D, ribosomes and chloroplast. Again, we mentioned earlier that chloroplast is for manufacturing sugar. Ribosome actually is also for the synthesis of a product, which is the protein. So protein synthesis is a function of ribosome during translation process. Now, we can therefore conclude that ribosome and chloroplast is the best suited group here, meaning they both have a function in manufacturing a product. So this is our correct answer now. Question number 19. Fatty acids are broken down initially by a process known as blank. So to answer this, let us do a process of elimination. Option A, glycolysis. We're already familiar with this process since this came from the word glyco, meaning sugar, and lysis, meaning breakage. So this is sugar breakdown. Again, glycolysis is a process of breaking down glucose into 2,3 carbon compounds called pyruvates or pyruvic acid. We can now eliminate glycolysis from our options since this involves sugar instead of fatty acids. Now let's go to option B, transamination. As the word implies, or as the name implies, it refers to the transfer of an amine group from one molecule to another. So transamination is a process by which amino groups are removed from amino acids and transferred to keto acids to form new amino acids. Further illustrate this, let us take a look at this chemical formula. So our amino acid alanine, and then we have your keto acid, the alpha ketogluteric acid. Then as what you can see, the amino group from the alanine is transferred to our keto acid, producing the glutamic acid. So you can see here, this is the amino group that was transferred. So we already know transamination, and we already realized that this involves amino group instead of fatty acids, so we can now eliminate this. Option D, from the word pentose, meaning sugar, we can already tell that this will involve sugar rather than fatty acids. But let us still take a look at the pentose phosphate pathway. In the pentose phosphate pathway, you have glucose 6-phosphate, and then its breakdown will produce ribulose 5-phosphate, which is a pentose. That's why it is called the pentose phosphate pathway. Ribulose 5-phosphate is actually very important part of the nucleic acid. Another importance of PPP or pentose phosphate pathway is its function or its role in NADPH production or synthesis. So what you can see in this process, you can generate NADPH in this process and in this process also. So again, just like options A and B, We can eliminate it now since this involves sugar rather than fatty acid. So we're now left with option C, beta oxidation. So let us further investigate what is beta oxidation. Okay, what now is beta oxidation? Beta oxidation, or fatty acid beta oxidation, is the process of breaking down a long-chain acyl-CoA molecule to acetyl-CoA molecules. So to see this, here is an overview of the process. So this is a cycle, and as what you can see at the end of each beta oxidation cycle, two molecules are formed. These are acetyl-CoA and an acyl-CoA that is 2 carbons shorter. Additionally, during beta oxidation, NADH and FADH2 are formed. So what's the importance of this? FADH2 and NADH are actually used by the electron transport chain to produce ATP. Actually, fatty acid beta oxidation is a key metabolic pathway for energy generation in organs such as liver, our heart, and our skeletal muscle. So during fasting, when your glucose supply is depleted or they become limited, beta oxidation is very important because most tissues, except our brain, can use fatty acids directly to generate energy. Okay, so going back to our question, fatty acids are broken down initially by a process known as beta oxidation, of course. Question number 20. Which of the following cellular structures is related to protein synthesis? Is it lysosome, nucleolus, mitochondrion, or the Golgi apparatus? So let us now put in a table their functions. So for lysosome, its function is to break down worn out proteins and organelles and other waste materials. So it's for recycling mainly. Mitochondrion is for energy production, that is why it's called the powerhouse of the cell. Golgi apparatus is for modification of proteins and lipids made in the endoplasmic reticulum and sorts them for transport to other sites, such as, for example, plasma membrane. And nucleolus is where rRNA or ribosomal RNA is transcribed and ribosomal subunits are assembled. So as what you can notice, this, the ribosome and the rRNA, these are important for translation or for the protein synthesis. So ribosomes, they are large macromolecular complex composed of rRNAs and ribosomal proteins that translates messenger RNA into protein. So they're important in protein synthesis. So the answer now will be nucleolus. They are the organelle that is important or related in the protein synthesis. Question number 21. The carbon dioxide produced by living organisms comes from the blank. So the chemical energy that organisms need comes from food. Cells do cellular respiration in particular to extract energy from the bonds of glucose and other food molecules. And then cells can then store the extracted energy in the form of ATP. So the answer here now is option B. Foodstuffs ingested by the organisms is where the carbon dioxide comes from. Now it's also nice to know where do carbon dioxide is released in cellular respiration. So carbon dioxide is released during the transformation of pyruvic acid or pyruvate into acetyl-CoA. That's what you can see here, carbon dioxide is released. And also, carbon dioxide is released during citric acid cycle. So, 1 carbon dioxide here and 2 carbon dioxide here. So there will be a total of 3 carbon dioxide produced per 1 molecule of pyruvate. And since there are 2 pyruvate in 1 glucose, so if it will be asked, how many carbon dioxide will be produced in 1 glucose, 1 molecule of glucose, it would be times 2. So 3 times 2 will be 6. So just like here in the chemical equation. Number 22. In pulmonary circulation in mammals, the veins carry blank. To answer this, let us review the pulmonary circuit in this diagram. Take note that the blue ones represent the oxygen poor or the CO2-rich blood, while the red ones represent the oxygen-rich blood. Okay, so once the oxygen-poor blood enters the heart by flooding into the right atrium via the veins, the inferior vena cava and the superior vena cava, then the oxygen-poor blood flows past the tricuspid valve into the right ventricle. The right ventricle then pushes the oxygen-poor blood past the pulmonary valve into the pulmonary arteries. These arteries will lead into the right and left lungs where the blood becomes oxygen-rich. Remember that the gas exchange occurs in the capillary beds of the alveoli within the lungs. And after this, the now oxygen-rich blood flows from the lungs back to the heart through the pulmonary veins, is what you can see here. The oxygen-rich blood will now go to the left atrium. Then it will flow past the mitral or the bicuspid valve into the left ventricle. Then, the left ventricle pushes the oxygen-rich blood past the aortic valve into the aorta. From there, the oxygen-rich blood then flows out of the heart and then spreads throughout the arteries of the body, as what you can see here in the systemic circulation. A useful mnemonic here is this. A for arteries, meaning they carry oxygenated blood away from the heart. Then V for veins, meaning they carry the oxygenated blood back to the heart. Again, arteries carry oxygenated blood from the heart. Veins carry the oxygenated blood back to the heart. So let us now take a look at the options here in our question. So for option A, oxygenated blood away from the heart is a function of, your clue here is away and oxygenated blood. So that would be a function of our aorta. Option B, oxygenated blood toward or back to the heart is a function of, so when we say toward the heart or back to the heart, it's a vein. And then as what you can see here, pulmonary vein is the one that carries oxygenated blood back to the heart. So the answer to our question is letter B, oxygenated blood toward the heart, is a function of our pulmonary veins. Okay, how about option C? It carries the oxygenated blood away from the heart. So this is a function of the pulmonary artery, right? It carries the oxygenated blood away from the heart to the lungs. Option D, it carries the oxygenated blood toward the heart. So that would be our inferior and superior vena cava. Question number 23. Which portion of the brain establishes regularity of respiration? So let us first take a look at the organization of the respiratory center in the brain. So the respiratory center is composed of several groups of neurons located in the medulla oblongata and the pons of the brainstem, as shown here in this figure. It is divided into four major collections of neurons. The first one is the dorsal respiratory group, located in the dorsal portion of the medulla, which mainly causes inspiration. The second one is the ventral respiratory group, which is located in the ventrolateral of the medulla, which mainly causes expiration but also functions in the inspiration. So these two are located here in the medulla oblongata. So the remaining collection of neurons we're going to discuss are both located now in the pons. So let us first discuss apneustic center. So apneustic center is located in the inferior portion of the pons or inferior pons, and it stimulates the neurons in the dorsal respiratory group, and it controls the depth of inspiration. Pneumotaxic center is located dorsally in the superior portion of the pons, or the superior pons for short, which mainly controls the rate and the depth of breathing. So again, Pneumotaxic center, it inhibits the activity of the neurons in the dorsal respiratory group, unlike the apneustic center, which stimulates the dorsal respiratory group. And pneumotaxic center controls the rate and the depth of breathing. We're gonna discuss more on pneumotaxic center because it was believed to be important in controlling the timing of the respiratory cycle. It is no longer considered essential for generation of the respiratory rhythm, unlike the dorsal respiratory group and the ventral respiratory group. But pneumotaxic center is involved in modification and fine control of the respiratory rhythm. So again, its main function is the control of rate and depth of the breathing. Going back to the question, which portion of the brain establishes regularity of respiration? So by looking at these options, we can obviously rule out cerebrum because cerebrum is not part of the respiratory center. Now, medulla, it contains again the dorsal respiratory group and the ventral respiratory group. Although medulla, the neurons of it generate the respiratory rhythm, it does not really control the regularity of the respiration. Now, we're only left with the pons. Is it the inferior pons or the superior pons? Again, inferior pons contains the apneustic center, while superior pons contains the pneumotaxic center. Between these two, the one responsible in the regulation of the respiration, or the one that establishes the regulatory respiration, is the pneumotaxic center. So the answer now here is option D, which is superior pons. Again, superior pons acts as the pneumotaxic center in the superior pons acts as a switch on and off of the dorsal respiratory group and ventral respiratory group. That's why it can control the rate and the depth of the breathing. Number 24. In the initial digestive process in man, all of the following occur in the mouth except. Okay, let us review the digestion that occurs in the mouth. When you eat, your teeth will cut and grind the food to convert it into smaller pieces. So option D, the conversion of food into smaller pieces, is actually correct. So as food is chewed, saliva, which is produced by the salivary glands, mixes with food. So what is saliva? Saliva is a watery substance secreted into the mouth by glands, providing lubrication for chewing and swallowing, and aiding digestion. The three major glands that secrete saliva are the parotid gland, the submandibular gland, and the sublingual gland. So what are the components of saliva? Saliva contains first mucus to moisten and buffer the pH of the food, and it also contains immunoglobulins and lysozymes to inhibit the growth of some bacteria and therefore prevent tooth decay. Also, saliva contains enzymes such as the amylase and the lipase. So, option A, secretion of enzymes actually occurs in the mouth also because it secretes the salivary gland secretes amylase while the tongue secretes lipase. Amylase is an enzyme that begins the process of carbohydrate digestion in the mouth, converting the starch or a complicated carbohydrate into a disaccharide called the maltose. The tongue produces lipase, which is another enzyme that begins fat digestion in the mouth. Both carbohydrate and fat digestion, the majority of their digestion actually occurs in the small intestine, but the primary digestion, the initial digestion of them, the partial digestion of them, can also occur in the mouth. So option B is correct. Carbohydrate digestion occurs in the mouth with the help of the amylase. Option C, fats and protein digestion. Although fat digestion partial fat digestion occurs in the mouth through the help of the lipase. Protein digestion does not occur in the mouth. The first step in protein digestion occurs in the stomach through the help of the proteases. So the answer here is option C. All of the following occurs in the mouth: the secretion of enzymes, the primary carbohydrate digestion, and conversion of food into smaller pieces, except fats and protein digestion. Question number 25. The growth reaction of plants to gravity is called blank. To answer this, let us first recall the definition of tropism. Tropism is a growth response of a plant toward or away from stimuli. So let us discuss here the stimuli involved in each of our options. So for option A, thigmotropism, it came from the Greek word thigmo, meaning touch. So it is a form of tropism in which the stimulus is touch. For option B, thermotropism, it came from the word thermo, meaning temperature. So it is a form of tropism whereby the plant displays a growth response or movement according to changes in temperature. For option C, heliotropism, it came from the word helio, meaning sun, so it is also called solar tracking. It is when a plant follows the movement of the sun during the day. The sunflower is the best example of a plant that displays this phenomenon. And lastly, geotropism, or also what we call the gravitropism, is a growth toward or away from the pull of gravity. So to guide you, here's a table that will show you the summary of what we've discussed. Again, thigmotropism, it's touch. Thermotropism, it's heat or temperature. Heliotropism is the sun. For geotropism, it's the gravity. So going back to the question, the growth reaction of plants to gravity is called geotropism.